Good morning, all. I just wanted to share a mini lecture which will help you see the format that we're going to use going forward. And it's about mitosis. So probably you all learned something like this in some biology class along the way.
Mitosis is a process of nuclear division by which replicated copies of a cell's DNA are organized into chromosomes. The identical copies of the DNA are then divided equally between two daughter cells. Early in mitosis, during prophase, the nuclear membrane and nucleolus begin to break down. As prophase continues, the DNA condenses to form chromosomes, while the mitotic spindle, made of proteins called microtubules, begins to form and attaches to the kinetochores on each chromosome. During metaphase, the chromosomes are moved to the central plate of the cell by the spindle microtubules. In anaphase, the microtubules attached to the kinetochores of each chromosome shorten, drawing the chromatids of each chromosome to opposite ends of the cell, while the unattached microtubules elongate, stretching the cell. During telophase, the chromatids, now called chromosomes, reach the poles of the cell as the unattached microtubules continue to elongate. The nuclear membranes begin to reform as the nucleoli reappear and the chromosomes relax as the DNA unwinds. Telophase is followed by cytokinesis, or division of the cytoplasm. In animal cells, a cleavage furrow forms at the center on either side of the cell, pinching through until the original cell is divided into two daughter cells. In plant cells, cytokinesis is different due to the presence of a rigid cell wall.
So first off, you can see they think all living organisms are plants and animals, at least all eukaryotes, which we don't. But the other thing is that, as you could probably imagine based on what you've learned so far in this class, that was a lie. And why it's a lie, I guess it's accurate for those organisms, plants and animals, but it is not an accurate representation of mitosis among eukaryotes. Now, what I want you guys to know is that among eukaryotes, mitosis varies in two different ways. The first is in the fate of the nuclear envelope, and the second is the position of the spindles. And I'm going to show you what that looks like. So first off, the location of the spindles in animals. The spindles are ortho, which means they come from straight on, like your orthodontist, whereas that's not always the rule. And the fate of the nuclear envelope in animals is that it vesicularizes. These are the little pieces of nuclear envelope. But that, again, is not always the rule. And what we see among eukaryotes is things like semi-open, where you poke little holes in your nuclear envelope, and that can either be ortho, straight on, or at an angle. And then these crazy closed mitoses, where the nuclear envelope stays intact, and you put your microtubules on the inside, or you put your microtubules on the outside and interact with your chromosomes through the nuclear envelope. So in the end, we can have semi-open pluromitosis, closed intranuclear orthomitosis. I don't want you to memorize these, but I want you to see the diversity, location of spindles, fate of nuclear envelope. And if we look at who has these, what we do in animals is that open orthomitosis, but we also find this in some green algae, acanthamoeba, some epicomplexans, some red algae, et cetera, et cetera. Semi-open, we find in some epicomplexans, some green algae, the oomycetes you haven't met. The pleuro is some other epicomplexans. Closed internuclear in these guys, closed extranuclear, including this crazy tunnels in some dinoflagellates. I want to just exemplify, I think, three kinds of this for you with some pictures. First, semi-open, little holes in the nuclear envelope, plural, not straight, in Plasmodium, the causative agent of malaria. Here is a cell undergoing mitosis in malaria, and if you take a minute and look for the spindles in this nucleus, the nucleus is on the inside. Maybe you can't see them yet, so I'll blow them up for you. Holy cramoly, there are 3 mitotic spindles in this section of the picture. 1, 2, 3. And what happens is there are little holes poked in the nuclear envelope through which the spindle is able to interact. And in this cartoon, we can see at least 5 mitotic spindles all at once. So this is not what animals do. And what this means for the Plasmodium is that it actually relates this mitotic property to cyclical fevers. So if we start with Plasmodium falciparum, here's temperatures right here. And so you might spike a fever for 2 days and then it drops, and then spike a fever for 2 days and then it drops. What's going on here is you have these little microsporidia. They invade red blood cells where they sit inside and basically take over the whole red blood cell. Now this is one cell with many, lots of genome in it, and then it goes through this rapid mitosis, chop, 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 chop into many little cells, splice and dice, and that's when the fever drops in falciparum. In vivax, it's something different. The fever spikes when the little cells invade and then drops and spikes when the little cells invade. But the point here is that cyclical fevers in malaria are associated with these mitotic, multiple mitoses at once. And here is a picture of what that looks like. Here's a red blood cell with the Plasmodium peeking their way out. A second example I want to draw out for your attention is closed extranuclear mitosis. So nuclear envelope stays intact, microtubules on the outside, and this is in an excavate called a parabasalid. These are the guys that digest wood in termites. And we'll just watch 2 minutes of this video here.
These are termites on a piece of wood. These single-celled organisms live inside termites. The question of how they got there has an interesting evolutionary answer. The story begins with plants. Plant cells have a cell wall that is composed of cellulose. Cellulose is composed of sugar molecules linked together into a chain. Recall that sugar is the primary food for animal cells. Any animal that could break apart the cellulose of plants into simple sugar molecules would have an advantage in the struggle for life. As it happens, no animal has yet evolved the necessary digestive protein. In other words, animals cannot digest cellulose, and so cellulose is not nutritional for them. This makes the termite diet something of a puzzle. For termites eat wood, and this seems to keep them alive. The solution to the puzzle is what you are looking at right now.
All right, so these are the so-called hairy man mastigoats, these crazy organisms that were in Lynn Margulis's article, where she argues this is the earliest example of a eukaryote, this kind of organism. They're excavates, but let's look for another minute.
The organisms inside the termite intestines do make the necessary digestive protein to break down cellulose. They break apart the cellulose into simple sugars to be used by the single-celled organisms as food. However, some of the sugar is released into the intestine and is absorbed by the termite.
All right, so you can, I hope, click on the link if you want to watch more of this crazy video about termites. The reason why these guys are important for us is that they have this unusual mitosis. They have these flagella at the one end of the cell, multiple flagella. And these multiple flagella are associated with this internalized microtubule structure that relates to the MTOC, the microtubule organizing center. And during mitosis, the microtubules interact. Here's the nuclear envelope. They interact with the chromosomes through the nuclear envelope. So this is extranuclear closed mitosis. And this is this thing called the karyomastodont that Lynn Margulis talked about, that I'm not sure other people have seen much evidence that this is a fundamental unit in eukaryotes, but here it is, closed extranuclear mitosis. And here's a SEM of this process as well. Here's the nuclear envelope, and these are the MTOCs that are going to be associated during mitosis. The last one that I want to exemplify for you is probably one of the coolest ones, which is closed extranuclear mitosis with tunnels in some, but not all, dinoflagellates. And I'm now going to show you the best image I can find of this crazy mitosis. And here it is. Yes, it's a crappy cartoon. Here's the nucleus, and somehow tunnels go through it, and the microtubules pass through, and the chromosomes attach. The last time people studied this, I think, is the 1950s, and so I can't answer any good questions about exactly how this works, but how weird is that? Instead, what I want to do is let's take the closed intranuclear mitosis. For example, this one that comes in at angles is plural. It's found in Microsporidia, which are fungi, forams, polycystina, and Cantheria, which are all rhizaria, some excavata, another excavata, and some amoebozoa. Is that monophyletic? No. Look at this, the one that we have animals. I already read this list to you. So if we take a phylogeny of eukaryotes, here's SAR, here's Archaeplastida, Amoebozoa, Excavanta. If we take these things and map them across, for example, the one that I pulled out there, the plural mitosis, extranuclear, we find that here and here and here and here and up in these guys here. So maybe some clustering, but then widespread. And here's what animals do. We have this open, and it's here, 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 and here. So it's quite variable among eukaryotes. And so that leaves two questions for the final. How does mitosis vary in eukaryotes? And the answer there is really the broadest level varies in the location of the spindles and the fate of the nuclear envelope. And the second question for you all to think about is, does the type of mitosis evolve rapidly among eukaryotes, or is mitosis very conserved? Finally, could you just drop me an email and let me know whether the quality of the video and sound here was excellent, sufficient, or unacceptable, not understandable? Thanks.